Hello and welcome to episode 6 of Evidence for Belief, a series which examines evidence for the existence of God. In this episode, we're going to continue to look at the concept of intelligent design within the living cell. Now, we're still in the realm of teleological evidence here, which looks at the purposeful design of something, which in turn provides evidence for an intelligent creator. In this episode, we're going to examine the third amazing aspect of living cells. We're going to glimpse the incredible world of molecular machines which operate inside all living cells and which keep them alive. The advent of the electron microscope has helped us to see that the living cell is teeming with literally thousands of what scientists refer to as molecular machines. These are microscopic protein molecules that move around inside the cell like tiny robots, performing all kinds of intricate tasks. Here are some snapshots from a video produced by Harvard University Biology Department showing computer-enhanced images of thousands of molecular machines at work within a single cell. Thousands and thousands of complex biological molecular machines moving around inside the cell. Now, we call them machines because they have machine-like characteristics, cogs and gears and motors, and many of them are like tiny robots with arms and legs. And each molecular machine is comprised of dozens or even hundreds of individual parts made of many different protein molecules, shaped and joined together to form all the various moving parts of the machine. And each machine is, well, they're alive. They're moving and living and doing things like little human beings. The living cell is a veritable factory filled with thousands of factory workers scurrying around doing all sorts of tasks. Some protein machines transport nutrients from outside the cell, through the cell wall into the cell. Some carry the nutrients throughout the cell and feed the other machines. Some transport waste back to the cell wall. Some repair other machines. Some build new machines. Some build the components of new cells. Some read the DNA instructions and copy bits of those instructions to take to other parts of the cell, a bit like delivery messengers. This incredible world of molecular machines that operates within cells increases our understanding of the irreducible complexity of cells. Because if any one of these machines or groups of machines was removed or was not in place in the beginning, the cell would not be viable. It would not be alive. All these complex biological components, this huge army of molecular machines, had to be in existence in the very first cells from the very beginning in order for those cells to be viable. In other words, in order to be alive and able to reproduce. Molecular machines could not have evolved incrementally over successive generations because the cells would not be viable. The ATP synthase machine is one of the more spectacular molecular machines that populate living cells. It has a motor and very intricate gears and cogs, and its motor spins at an incredible 130 revolutions per second. If you happen to see a life-sized version of this machine operating in a factory, you would definitely conclude someone with intelligence has made that. You certainly wouldn't conclude that it simply formed itself out of a primordial swamp. Biologist and author Dr. Alex Williams explains, the ATP synthase machine is a proton-powered motor that produces the universal energy molecule ATP, adenosine triphosphate. When the motor spins one way, it takes energy from digested food and converts it into the high-energy ATP. And when the motor spins the other way, it breaks down the ATP in such a way that its energy is available for use by other metabolic processes. Now, this is just one of thousands of molecular machines that operate within a living cell. There are molecular machines for converting energy stores to usable nutrients, for transporting those nutrients around the cell, for removing waste, for repairing damaged molecular machines, for breaking down dysfunctional molecular machines and recycling their parts, for making new molecular machines. The cell is literally swarming with thousands of specialized molecular machines. For example, There is the extraordinary assembly line of molecular machines within the nucleus, at the very heart of the cell, responsible for producing new proteins. There are molecular machines to uncoil a strand of DNA ready for transcription or copying. There are RNA polymerase machines to transcribe or copy a section of DNA into messenger RNA. Then there are molecular machines to cut and splice sections of this mRNA. Then there are molecular machines to transport the mRNA outside the nucleus into the cytoplasm of the cell. There's a molecular machine known as ribosome, which binds to the mRNA and reads the code in the mRNA to produce a chain made up of amino acids. Then there are transfer RNA molecular machines to link new amino acids to the mRNA chain in the correct order, thus forming a new protein. There are molecular machines known as chaperones, which then fold the new protein into its correct shape. Then there are molecular machines to carry this new protein to the site in the cell where it is needed. And all of these machines are essential to the proper functioning and survival of the cell. And without all of them working together, the cell would die. So, in this sense, the humble cell can be said to be functionally irreducibly complex. Its viability, in other words, its survival, and its ability to replicate is absolutely dependent upon the simultaneous coexistence of a vast army of interdependent machines. These molecular machines had to be in existence and fully operational in order for the first cell to be alive. So in this sense, the living cell is irreducibly, functionally complex. Furthermore, each of these molecular machines individually is also irreducibly complex and cannot have evolved incrementally and gradually. You see, each molecular machine is comprised of many individual interdependent moving parts made of different proteins, folded and shaped and joined together in just the right order to construct the machine. Here's a list of just a few examples of molecular machines within a cell and the number of proteins that each one of those machines is comprised of. Eukaryotic ribosomes are machines which copy segments of RNA and synthesize new proteins. They are comprised of 80 different proteins. The 26S proteasome is a machine which deconstructs damaged proteins, a bit like a garbage disposal truck in the cell, and it has 66 protein components. The NPC, or nuclear pore complex, is a machine that transports newly formed proteins and other molecules from the cell's nucleus to the cytoplasm. It has 1,000 individual protein components. The spliceosome is a machine which splices RNA in the formation of new genes. It has 300 individual proteins. The centrosome is a machine which oversees some important functions during cell division, and it has 500 individual protein components. Now, there are literally millions of different types of proteins that could be used to construct molecular machines, but only the correct specific proteins assembled together in just the right order and in just the right shape must somehow be selected and joined together to make each molecular machine. So going back to the spliceosome, if any one of its 300 component proteins was missing, or was in the wrong order, or was a different protein altogether, if just one of the millions of other possible proteins was included in the structure at any one of those points, the molecular machine would not function. All the right proteins in exactly the right order have to be in place for the molecular machine to function. In this sense, molecular machines are irreducibly complex. They cannot have evolved incrementally over time because a partially constructed machine would be non-functioning and would have no evolutionary value. And non-functioning molecular machines would, in turn, render the whole cell non-functioning. So the cell itself would not be alive and able to reproduce. But the irreducible complexity of these molecular machines is made even more extraordinary if we zoom in even further and we understand that proteins, the building blocks of these molecular machines, are themselves comprised of hundreds of different amino acids, each of which is arranged in precisely the right order and shape. You see, the average protein is comprised of about 400 different amino acids. And only the right amino acids joined together in exactly the right order will form a functioning protein. And each of the millions of different proteins has a completely different sequence of amino acids. And as we've seen in a previous episode, the statistical chance of even a single protein forming by random chemical combinations of amino acids is ridiculously improbable to the point of statistical impossibility. There literally haven't been enough seconds in our universe's existence for a single protein to have formed by chance. In a previous episode, I mentioned a quote by Dr. Thomas Hines, Professor of Organic Chemistry at Friedrich Schiller University, Jena. In his article, "Did God Create Life? Ask a Protein." It's worth stating again. He writes, "Proteins are so complex, they'll not form anywhere in nature except in living cells. Inside cells, the directions for protein construction are already contained in the DNA. Then, if a protein is to perform its task, its production must be carefully regulated. But even then, it will not function unless it also has the correct address tag and is properly folded. All these systems would have to have been in place, or the first cell could not function. These systems, however, are just the tip of the iceberg. I chose them to illustrate the many coordinated systems that would have to have been present before the first cell would work." He goes on to say, "The teaching that the first cell spontaneously popped into being without the involvement of the Creator has its basis in the pre-scientific myth that single-celled creatures were simple. It obviously does not stand up under today's knowledge that a cell's DNA, RNA membranes and proteins are extremely hard to make, and when proteins are made, they must be properly folded, addressed and turned on and off at just the right times. None of these brilliant solutions could invent itself, yet no first cell could exist without all of them. They could not have happened without a very intelligent Creator." Dr. Sir Fred Hoyle, Professor Emeritus of Astrophysics at Cambridge University, expresses the same thought. "The chance of obtaining even a single functioning protein machine by chance combination of amino acids is equivalent to trillions of blind men all solving Rubik's Cube simultaneously." Biochemist Dr. Michael Denton, in his book Evolution, A Theory in Crisis, states, "We now know that there are in fact tens of thousands of irreducibly complex systems on the cellular level. Specified complexity pervades the microscopic biological world. Although the tiniest bacterial cells are incredibly small, weighing less than 10 to the minus 12 grams, each is in effect a veritable micro-miniaturized factory containing thousands of exquisitely designed pieces of intricate molecular machinery made up altogether of 100,000 million atoms, far more complicated than any machinery built by man and absolutely without parallel in the non-living world." But zooming back out from these biological building blocks of amino acids and proteins to look again at these molecular machines as a whole, there's also a profound mystery to them. What makes them work? What makes them move? You see, it's one thing to join all the right amino acids together and then all the right proteins together and join them together in exactly the right order and fold them together into exactly the right shape to construct these molecular machines, but what actually makes them come alive? What makes them move and function? How do they know to do what they do? How do they know to go where they need to go and perform the intricate tasks that they perform? What brings them to life? Now, no scientist can actually answer this at any depth. There is an incredible mystery here. Now, in order to try and explain this, scientists have coined the term autopoiesis to refer to this self-life, this self-perpetuating, self-organising, self-repairing, self-replicating life force that seems to exist within cells as a whole and within each of these molecular machines individually. Biologist Alexander Williams believes that autopoiesis makes a stronger case for intelligent design than even irreducible complexity, because it is inherent in all biological life. Listen to what he writes. He says, autopoiesis provides a compelling case for intelligent design in 3 stages. 1. Autopoiesis is universal in all living things, which makes it a prerequisite for life, not an end product of natural selection. 2. Autopoiesis is not reducible to the laws of physics and chemistry. In other words, it cannot be explained by mere naturalistic processes. And 3. There is an unbridgeable abyss between the dirty, that is, inefficient, mass action chemistry of the natural environment and the perfectly pure single molecule precision of intracellular biochemistry. What he's saying is that there's a great mystery here. Despite coining a term for this self-life of molecular machines, calling it autopoiesis, we really don't understand it. Merely giving something a name doesn't explain it. And there is no natural explanation for this. Something is going on here at a profound level. There is a life force that we cannot comprehend and cannot explain, and which only makes sense if we are indeed the handiwork of an all-powerful, life-giving, intelligent designer. Professor Dean Kenyon, a world-renowned biologist, was originally an atheist and an evolutionist, and as an atheist scientist he wrote the acclaimed textbook Biochemical Evolution, arguing for how the cell could have evolved by chance processes. But after the invention of the electron microscope and the discovery of these molecular machines within our cells, he was converted to the creationist viewpoint. When he first looked down the barrel of an electron microscope, or perhaps, to express that better, looked at the screen on the computer from an electron microscope, his atheism was destroyed, because he realised that this living cell, teeming with complex molecular machines, could not have evolved incrementally by random chance processes. In a video interview after his conversion, Dr. Kenyon stated, "Nothing short of an intelligence could have created this intricate cellular apparatus. This new realm of molecular machines is where we see the most compelling evidence of design on the earth." The microscopic world of the living cell, with its complex, gross physical structures, as we saw in a previous episode, its amazingly complex DNA encoding, which we saw in the previous episode, and its plethora of extraordinary living, moving molecular machines, all of that cries out for an intelligent designer. In fact, I think it takes much more faith to believe that all this came about by random natural processes rather than by the design of an all-powerful intelligent creator. The simplest and best explanation for the living cell is that it was marvellously designed and created by an all-powerful creator God. World-renowned biologist Dr. Michael Behe, in his book Darwin's Black Box, The Biochemical Challenge to Evolution, wrote this: "Darwin knew his theory of gradual evolution by natural selection carried a heavy burden. He stated, if it could be demonstrated that any complex organism existed which could not have been formed by numerous successive slight modifications, my theory would absolutely break down." Dr. Behe then argues that the irreducibly complex living cell, with its irreducibly complex molecular machines, has surely met Darwin's criteria for the failure of the theory of evolution. And so he writes, "As the number of unexplained, irreducibly complex biological systems increases, our confidence that Darwin's criterion of failure has been met skyrockets toward the maximum that science allows." I agree. The living cell cannot have evolved via incremental, random natural processes. It cries out for the existence of a supernatural creator. For more information on any of these topics, you can check out my books, which are available on my website, smartfaith.net, where you can also subscribe to my weekly blog. Thanks for watching episode 6. Please check out episode 7, and in the meantime, have a great day.